Hi, I'm Nicholas Marchanda. Here's the fun, the cool guy physiology review right here. And so I'll first start off talking about homeostasis, right? Homeostasis is where your body feels comfortable, right? So a popular example of homeostasis is body temperature, right? There's a specific temperature we have to be, and if we're not that, we're probably dead. And so I'm going to just call your average body temperature 98 Fahrenheit. Might not be 100% true, but for the purposes of this explanation, let us assume that this is your resting body temperature, where your body is ideal at. That is your set point, this dashed line right here. And your body has these tools that make sure it stays within this range. This range is homeostasis. It won't ever, or 90% of the time, be exactly at 98, but it's close enough where the cells in your body can thrive and where you can, live and grow, right? This is comfortable. And your body has different solutions and problems occasionally of making sure you stay in homeostasis. And so it does this by having a sensory motor reflex pathway, right? Where it can detect these changes when it's going too high or too low, and then can course correct to make sure it's still in that range. So first, your body needs a stimulus to react to, right? So let's say it's hot outside. Your sensory neurons will detect this change in temperature. Your sensory neurons will then send this to the afferent pathway, which sends information to the brain or some other integration control center. This can occasionally mean the spine. You'll see me lean in and touch the computer to make sure it's still on and working at times. And so this afferent pathway will send information to the control center or brain. I'll just write control here. But at times, the control center can be the spine. Don't forget that. Then we have afferent pathway. Because after your brain comes up with a solution, like whether it's taking off clothes because they're too hot or sweating, it needs to send out that information. So it sends out that information through effector nerves, right? And can you still see it? Okay, barely. It sends it out to the effectors. Perfect. And so let's run through an example of this just to make sure we have this down. Stimulus, you're running outside and it's hot, like 90 degrees hot. And so your brain, your nerves, I should say, detect this change in temperature. And it's like, okay, we need to do something about this. Sends this information away from the nerves towards the brain so the brain can think of something. The brain starts to get this information. It's like, all right, I think we should just start sweating, right? We're moving. It'll be an effective tool in order to reduce our temperature. Because right now we're sort of starting to break out of homeostasis here. It's not going too hot. So it sends out that information to the efferent neurons. And then after that, it gets to the effector sweat glands. And then you start sweating, and then your body begins to return to homeostasis. So that of itself is a perfect example of a negative stimulus. So it's a negative stimulus. Here, I'll get out of your way here. Perfect. All right, a negative stimulus is when the problem causes the solution to prevent more of the problem, right? Just like how when the problem of heat causes the body to come up with a solution of sweat, you sweating most of the time will not cause you to get hotter. You're not going to sweat forever, right? You'll get hot, you'll sweat, then you'll get cool and you'll stop. It's when the feedback of the cooling down stops the problem from growing, right? Like if this was a positive feedback loop, this would just go on forever if the sweat caused you just to get hotter. But thankfully it doesn't, and we stay cool and not dead. A positive feedback loop is the Opposite, right? You have the problem. The problem causes the solution to cause even more of the problem. And you might be thinking to yourself, Nick, that sounds terrible. How can that ever be a good thing? And I'd say 90% of the time, you're right. It's just terrible. Usually if you have a positive feedback loop going on, you also have a really big problem. Fortunately, there are a few situations where this is not even encouraged, but necessary. Like, let's say childbirth, right? So mom has a problem. She has an 8-pound kid coming out of her. That causes a lot of pressure and pain on the cervix, right? This pain causes oxytocin to be released, and this oxytocin goes to the brain, the pain, sorry, I messed that up, my wording. The problem is the pain, right? The pain goes through the brain, and the brain's like, well, we'll just release oxytocin, right? And that's the solution, the oxytocin. The thing about oxytocin is that in the cervix, it causes contractions, which causes the baby to be pushed forward more. And that causes even more pain for mom. You're probably thinking that sucks for mom. And it does, because it just leads to this positive feedback loop where she's contracting, she's pushing the baby out, which is necessary, but it hurts and hurts and hurts, which is unfortunately part of the solution, which the oxytocin gets released. So there's more contractions until the baby's out. It's a positive feedback loop. Eventually, the baby is out. There's no more pressure or pain on the cervix. And once you have no more problem, you have no more need for solution. And so that's just the groundwork of positive feedback loop. Sort of sucks for mom. A little uncomfortable maybe. I wouldn't know. And so that's the groundwork for homeostasis. I'll erase this. Now let's talk about something a little more robust. Perfect. All right. So we're going to be talking about different kinds of protein-mediated transportation in the cell. First, we'll go for the simple, like channel diffusion, right? I'll use an iconic example here. The water pore in the cell, right? Because the outside of the cell has these hydrophobic lipids or whatever. It's not going to let water in. And it's exactly what it says on the tin, right? It's just a channel. It diffuses, right? It's super quick, right? Because the protein doesn't change shape, right? It's just like a tunnel. And it is pretty specific, right? So it'll just rush in or rush out according to whatever osmosis tells it to at the time. And it's specific. Usually competition isn't too big of an issue because it's just so darn fast, right? Like there's no like checks or balances here. You're in or you're out. It's not much of an issue. And usually it's like compared to like a graph like this, like greater the difference, greater the rate of change. Like it's 1, 2, right? It's not as complicated as something as interesting as facilitated diffusion, right? So facilitated diffusion's a little more complex, right? It still works on the basic principle of diffusion, right? But I like to imagine it like this. Sorry, I'm not good at drawing. Let's just like imagine. This is the reason why I'm in STEM and not in like liberal arts or something. I don't have that talent. Perfect. And so these guys are also incredibly specific, right? A certain molecule, we'll call it molecule O, will float around, fit into it. As the molecule gets into the facilitated diffusion, right? This will change shape and eventually let it go through. So let's talk about how specific it is. The answer is very, right? It's only going to let this kind go through. It might let something else go through that's incredibly similar, but, if you're that unlucky, I'm sorry. Yeah, and it reacts to different stimuli to function, right? You have, I believe, electrical, chemical, pressure, stuff like that. But it's facilitated, right? And there is a limit to how effective it can be, right? Like there's only so many of these on a cell, right? It's not like simple facilitated where it's just essentially a hole in the wall you can walk through. Like it takes time, right? And it can only be as effective as the number of these, I don't want to call them chambers are, right? Like if you have 10 of these, it's only going to be as effective as 10, right? It can't get any more effective past that point. And so that leaves us to our last point. Of active transport. All right, so active transport is unique because unlike all of the past stuff I've talked about, it works against nature, right? And it requires ATP or GTP to function. So let me give you a really popular example, all right? I'm a big fan of this one because it keeps me not dead. We have I believe it looks like this. You have to forgive me. Essentially the way it works though, it's you guessed it, sodium potassium pump, right? Pumps 3 sodium out and 2 potassium in. This is a big deal, right? Because one, this is where not the majority, I can't say that for certain, but where a lot of our energy goes in order for our cells to maintain that artificial gradient. And it enables something called secondary transport as well. And, okay, sorry. So secondary transport is a little unique. Active transport is obviously this, right? You have the little slot for the ATP or whatever, where this is actually happening. This is the active transport, number 1. Then you have the secondary transport, number 2. And so let's say there's a channel here or something. And so because of this artificial gradient we've created where there's way more K in here than K out here, K will naturally begin to slip out. However, your body has designed it for some channels where as potassium is leaving, something else, I don't know, I'll just call it X, will leave with it. And this is only possible because of the artificial gradient that we've created. I will call this, I ain't calling it anything. This is just what it's called, symport. When 2 things move out in the same direction due to the artificial gradient provided by our number 1 active transport. The second kind is, as you've imagined, the opposite. As potassium goes to leave, element or molecule Y goes and enters. This is called an antiport. So as for specificity, to my understanding, this is quite specific, and this process is as fast as the gradient determines it, all right? Like if your cell isn't working hard to change this or create that gradient, then the secondary transport will be all but shut down, right? So yeah, I believe that is everything for the second part here. And now we get to talk about the final, yet certainly not any less interesting aspects of osmolarity and tonicity. So, how should I start this off? We'll start off with osmolarity versus molarity. So these things, well, they look the same, certainly aren't. So osmolarity is number of moles per liter. Osmolarity is number of solute particles per liter. And they sound similar until you consider the implications, right? So let's say you have 1 mole of NaCl, classic salt, right? And like this is OChem 1 stuff. However, if you put this salt in water, this will break apart, right? And so you'll have 2 osmoles when a salt is dropped. And so this principle of things breaking apart in water is incredibly important to the human body, right? Like you can imagine the importance of being able to understand this. All right. So we'll have our osmolarity here, and then we'll have tonicity here. So let's run through a few basic principles of osmolarity and tonicity as well. Osmolarity is just the numbers, right? It's just what is. Let's say we have our cup of water. Let's say it's 300 milliosmoles or whatever. Then you have your cell, which is 100 milliosmoles. We would look at this and we would properly say, well, the solution is hyperosmotic to the cell because it's just more concentrated, right? So we call this hyperosmotic. And likewise, tonicity is unitless. Tonicity doesn't care what is, it just cares about what happens, right? In the same solution, it would be hypertonic because using the principle of osmosis, water goes to where the party is, right? And if there's more party outside of the cell or more particles, I am proud of that one. I thought of that on the spot. You better take notes. Water goes to where the party is, the particles. That was tight. I'm actually genuinely excited going through that now. Holy Hannah. All right, back on track. Jiminy. It goes to the particles, right? And as a result, the cell is sad, it's shriveled, it loses its water, and probably dies. Right? So you can see why doctors would be really passionate about this sort of stuff, because if you mess this up, the patient can and will die. And so, looking at this example I provided here, you might be tempted to say that, oh, hyperosmotic always means hypertonic. That is not true. Let's bring in a different example here, right? Because we have to consider the permeability of the cells. So let me think of one right now. There's one good one on the lab. All right, let's do this one. So we have the cell here. The cell is, happy as a clam, right? And so we add 5% dextrose, all right? So this 5% dextrose is roughly equal to the 300 milliosmoles in the cell. And so initially it's isomolar, right? It's equal. So this is isomolar. However, cells have this interesting trait of literally eating everything they can. And it can and most definitely will eat this dextrose up. So nom, time passes. And so everything's been eaten up. So now what happens? Well, you've just added glorified water to a cell, my friend. And the cell is going to be where the particles are. So the water rushes in. It's like, man, it's so lonely out here. What happened to all the glucose? I don't know. But there's more party in there than there is in here. And so it rushes into the cell, becomes hypotonic, which is where, I'm spelling this right, which is where the cells, well, not the cell, my apologies, the water rushes into the cell and it inflates like a poor child's balloon and eventually pops, right? And so that's just like one example of the differences between osmolarity and tonicity, right? You can do another example where this is off the cuff, so you can do another example where, say, you have a cell and it's full of this impermeable, of its impermeable parts, right? And you have surrounding it like these very permeable parts. I think urea is permeable. We'll run with that. Oh, you can hardly see my green dots. I'm sorry. Here, let me bold these for you. Perfect. Okay, now you can see them. So technically on the surface, they're both equal, right? Both have four, well, we'll just call 4 milliosmoles, 4 dots, 4 milliosmoles. 4 milliosmoles on both parts, so it's isomolar. But since these are impermeable, but these are, they're going to want to hit their own kind of equilibrium. And they'll rush in there to hit their equilibrium, right? 2 green on the outside, 2 green on the inside. Molecules and particles think for themselves, right? They don't give a flying flip about what's already in the cell. And because of that, you're going to end up with this situation where, can you see this? Okay, you can. There's more on the inside of the cell than on the outside cell. And where does the water go? It goes where the party is, where all the particles are. And all the particles, they're in the inside of the cell. Water wants to rebalance that, rushes in. Isomolar, hypotonic. You can't see that, but trust, it's hypotonic. And yeah, that's a long and short of it. Let's see if I missed anything. I don't think I did. Thank you for watching. And to the poor TA or tutor that has to watching me yap nonstop, thank you for taking the time to watch this. If I missed anything or screwed up in any way, let me know. Thank you for your time, and I hope you all have a wonderful day.